Okay, video 2, going into cell respiration and fermentation. Um, this will likely be a 2-part video. Gonna try and keep it at about 10 minutes. So let's get in. Whoops. So first thing we need to talk about, sorry, I'm glancing at my phone. I'm setting a stopwatch so I can keep track of the time. So a theme in respiration is going to be these electron carrier molecules. Because the general idea is that as we break glucose down, we're actually going to be breaking it down in a series of steps, small steps. And with each of those steps, we're going to get high energy electrons, just regular old electrons, just with some extra energy. And they're going to be given to these two types of molecules, NAD plus and FAD. They're both just coenzymes. Their job is to carry electrons to the electron transport chain and then donate them to oxygen, where it'll form a water molecule. So these will show up throughout this lecture. Whenever you see them, just think they carry electrons, and you can think of these electrons as little bits of energy, less than ATP. We're going to use all those electrons to make ATP. So this is the general flow. We talked last time about exergonic versus endergonic reactions, and this is an example of a pairing up of one. So we take an exergonic reaction, which is the stepwise breaking down of glucose, and we get all those little electrons from it, and those electrons get carried by NAD plus to the electron transport chain, where they are used in an endergonic reaction, which actually makes ATP. The actual reaction to make ATP is endergonic, so to do it, we have to pair it up with exergonic reactions to get the energy needed. So the energy is carried by those electrons, and overall, the overall process does go downhill. It is overall an exergonic reaction. It just has a few endergonic steps in it. Okay, so this is the very general overview of cellular respiration. We're going to be talking about each of these steps in more detail, starting with glycolysis, moving into pyruvate oxidation, then the citric acid cycle, and finally ending off with oxidative phosphorylation, which is the electron transport chain, and chemiosmosis. This lecture will probably just cover glycolysis through the citric acid cycle. So, glycolysis. As we go through these, there's going to be a lot of names thrown at you. You do not need to memorize any of the enzyme names. You don't need to memorize individual steps. What you need really is the net, what's in that net box down there. That's kind of the important stuff. But in general, glycolysis is the first step of respiration. It happens in the cytoplasm, so not in the mitochondria. It's the only step really that is not in the mitochondria, and it is kind of split into two phases. The first is an energy investment phase, where essentially you use a little bit of ATP to destabilize the glucose molecule. You use that energy to get the bonds a little wobbly so that you can break them later. And that happens actually in the payoff phase, where we're actually going to form ATP and convert our glucose molecules into pyruvate. Pyruvate is one of the names you should know from this. It is an intermediate molecule, but it is where most of our energy is left. Additionally, in the process of making the pyruvate, we did make 4 ATP, and we gave 4 electrons over to NAD+, so we have some energy electrons that are going to be carried forward to the electron transport chain. All in all, what we've done, we've taken glucose and we've turned it into pyruvate and a little bit of water. Next, what else have we done? We have made a net of 2 ATP. We made 4, we used 2, so overall we had a gain of 2. And then lastly, we had some electrons that are getting carried to the electron transport chain later. Don't worry about the numbers of electrons or the specific numbers of ATP. Just keep in mind a general flow. So like in general, glycolysis produces a little bit of ATP. It gives us a little bit of high energy electrons, and the rest of the energy is stored in that pyruvate molecule that will continue through the process of respiration. Every single organism on this planet does glycolysis. Every single one. It is a big piece of evidence for the common ancestry of all life. Did I ever hit record? Oh, totally did. Okay, sorry, just had a moment. Okay, so that's glycolysis. We'll kind of wrap around back to this when we talk about fermentation, but for now, let's get into the mitochondria. So, the mitochondria has, as we hopefully know by now, one of its main things is that it has distinct compartments. It has the intermembrane space and then the matrix itself on the very inside, the yellow. And that lets it have different chemistry in the two compartments, so that you can essentially what you're going to be doing is establishing a concentration gradient that we'll use later. It also has its inner membrane with the folds called cristae. We do the folding to increase our surface area so that we can fit more enzymes and proteins that'll actually be doing cellular respiration on them. So, the next step, the citric acid cycle, also paired up with pyruvate oxidation. I don't show it here because there's not really too much to know. Do I want to connect me labs? Yes. But pyruvate was the product of glycolysis, and before it enters the citric acid cycle, it gets converted to acetyl-CoA. In the process, we get some carbon dioxide and NADH, again carrying some high energy electrons. So just a quick little conversion. We get a little more energy out. It happens in the mitochondria before it gets to this citric acid cycle. So let's talk about the citric acid cycle. The main goal of the citric acid cycle is to take acetyl-CoA, which is a 2-carbon molecule. CoA is a coenzyme A. Don't need to worry too much about it. But the first thing that happens is we take our acetyl-CoA and we react it with another molecule, oxaloacetate. You don't need to know that one. But we make citrate, which is a 6-carbon molecule. And that is citric acid. That's where it gets its name from. So citrate is the thing we are going to be breaking down. Essentially, the next few steps are going to be taking off a carbon from citrate. It's going to be converted to carbon dioxide. And in the process, we're going to get some high energy electrons that will get carried forward in this process. So we see it goes from citrate, to this 5-carbon molecule. We get some NADH and CO2. We break it down again. Again, we get some NADH carrying those high energy electrons and CO2. Now we're down to a 4-carbon. This is where it will stay. The next few steps are really just converting that 4-carbon product back into oxaloacetate, which was used at the start to react with acetyl-CoA to make citrate. So that's why it's a cycle, because it regenerates oxaloacetate every turn of the cycle. And it just needs a new acetyl-CoA to actually do one revolution. So in this whole process, what have we done? We have taken our acetyl-CoA, which was 2-carbon, and we have removed 2 of those carbons and converted it to carbon dioxide. So this is where the carbon dioxide of respiration really comes from. And in the process of doing that, we have captured some high energy electrons, about 4 of them, 4 pairs, I should say. We've also made an ATP, just by chance in the process. We happen to be able to get an ATP. You'll see in the diagram, it's called GTP. They're the same thing effectively. The difference is instead of adenosine triphosphate, it's guanine triphosphate. So still kind of based on a nucleotide, still the same purpose. And then finally, yes, we did regenerate the oxaloacetate. So the purpose of this is removing the energy in little bits, releasing CO2, and capturing that energy in the electrons that get brought forward to the electron transport chain. which is where we will begin the next video. Let me know if you have questions, and that's all.